last speaker of the session, and that is Dr. IL51 Gacche from Department of Biotechnology, Savitribai Pune University. And his area of interest includes natural product and drug discovery, cancer biology, diabetes, bioinformatics, and
thank you so much. Just let me, my screen is full share or half share? Is it full now?
Yes, it is.
Hello? Hello?
Yes, you are audible and you are
I will start. So good morning to all of you. At the outset, let me apologize myself for the inconvenience or to the organizers, which I promised them to be there, but unfortunately, because of some technical difficulties, I could not make it. Today, I will be having a very cursory look at what we have done for this, the role of dietary flavonoids in regulation of epigenetic markers, along with those microRNAs which are also involved in epigenetic processes in triple-negative breast cancer. I hope my slides are moving. So this is, I just wanted to have a refreshing awareness to the audience that many of you may be knowing that these flavonoids is a very interesting biologically active group from the nature, and multiple biological activities have been assigned to this particular scaffolds. Around 7,000 or more than that even flavonoids have been identified from different resources and have been annotated with different biological functions. Recently, they have also been known to regulate the functions of microRNAs, the circular RNAs, and all those small non-coding RNAs, which per se were not earlier known to have attributed with the functions of these flavonoids. We all know that the recent literature has accumulated which relates the functions of flavonoids with lot of cancer cell signaling pathways. They downregulate the proliferation, upregulate the apoptosis process, inhibit the migration, invasion, and also they are recently shown to have some effect in amelioration of emerging drug resistance. And surprisingly, when I was browsing this clinical trials GOV yesterday, there are around 178 clinical trials. People are either trying to use these particular molecules as adjuvant therapy, or they also are interested to link this molecular biological activity of these molecules for circumventing the drug resistance, and to a greater extent also for the off-target toxicities in cancer management. So when we talk about flavonoids and how they regulate the epigenetic frame landscape, that there are some 3-4 aspects that we need to understand. The first is they are known to inhibit these all enzymes which are involved in epigenetic regulation of the cancers. They also have either they activate the tumor suppressor genes or block the suppression of oncogenes. These particular molecules are also reported to repress the non-coding RNAs which regulate the chromatin structure and aberrant gene expression. And we also know that these molecules also works in methylation and demethylation by inhibition of this DNA methyl transferase and so on and so forth. So it was actually a project given by the SERB, and we set these 4 objectives to work on this. We had a structurally diverse panel of the dietary flavonoids, and we wanted to see whether how these particular flavonoids regulate the microRNAs, and what sort of differential expression patterns do they exert on the oncogenic and tumor suppressor microRNAs, which per se are related with the functions of epigenetic functions. We also had opportunity to test the combinatorial effect of these promising flavonoids with the standard drugs, and we were interested to know the structural requirements for effective inhibition of these epigenetic regulators by building a 3D QSR model. So this is a framework of methodology that we have adopted. We first of all kept on screening the flavonoid molecules for the entity assays, and then scratch, avoids, that set of parameters that we usually test for the efficacy of any biological molecule, that we wanted to ascertain that these molecules have effect on anti-cancer mode. So we have also this panel of HDAC 1 to 11 isomers. We tested all the isomers with this limited test of, we can see how it regulates the AMPA and tumor suppressant. I will let you know, and we also tested some of the ABC efflux transporters, MRP1, PGP, and we also initiated the efforts of cloning the promising tumor suppressant microRNAs in our own plasmids and see for its functional validations. We have also performed the protein expression profiles by using Western blot, and we did some molecular docking and molecular dynamic simulation for asserting the structure-active relationship. So this is, these are my two students, Snehal, which happens to be a PhD student, and Dr. Navnath, who is a postdoctoral, have done this entire work. We initially started with testing the set of flavonoids, some 10 molecules. Actually, we have 10 molecules by in silico screening on various targets of the anti-cancer, and we wanted to ascertain that the test molecules have activity that we have procured from the commercial sources, and we wanted to establish, yes, they do have the biological activity. And we then calculate the IC50 values, and you will also see that some of the impressive molecules like galangine, luteolin, and ricin, camphorol, they have adverse effect on the morphology of tumor-negative breast cancer cells. We have used MDA 231 as a model cancer cell and for entire study. We went ahead and we also wanted to see whether these molecules have ability to inhibit the migration, and do they have impressive effect on the inhibition of the migration of the cells. We performed the scratch assays, and if you look at that, the standard drug level called as SAHA is epigenetic, HDAC inhibitor. As compared to that, there is significant reduction of the wound closure. We know that developing the anti-cancer strategy by inducing the ROS is not a new strategy at all. For example, these drugs, cisplatin, doxorubicin, 2-methoxystradiol, and these all drugs, they do induce the ROS in the cancer cells and help demonstrate the cytotoxicity and kill the cancer cells. So we have also checked, we have taken some promising molecules like apigene, galangine, and luteolin, and we see that they do have the ROS inducing capabilities in the triple-negative breast cancer cells. You will find that apigene has demonstrated significant effect in inducing the ROS that we tested by DCFD dye. We went ahead and also seeing some whether these molecules have any effect on the arrest of cell cycle in triple-negative breast cancer, and you will find that as compared to the standard drug, they have significantly arrested the cells at G0, G1 stage. We further went ahead and we have seen that how effective they are in inducing apoptosis in the cells, because we know that apoptosis inducing capability happens to be one of the strategies for developing the novel anti-cancer candidate drug molecules. And here you will find that these promising flavonoid molecules have significantly accumulated the number of cells which are undergoing in apoptosis. But we can see in quadrant Q2 that there are number of cells which are undergoing the apoptosis. Then we have selected a panel of 11 HDAC molecules, which our main intention was to see how differentially these flavonoids regulate the expression of these all HDACs, because these HDAC molecules, although they have some consensus sequences in their active site, but they do have different physiological functions. And here you will find that these different molecules have differentially exerted their effects on the regulation of the HDAC molecules. Here you will find that the most important HDACs which are in concert with regulating the progression of the TNBC cancer are HDAC 1, 3, 9, and you will find that here these apigene and galangine molecules, they do have very impressive effect on down-regulating the expression that we have analyzed by using qPCR. You will also find that these molecules are not effective against DNMT3A, DNA methyltransferase, but they do have effect on DNA methyltransferase 1, which is involved in transfer of methyl groups in the methylation process. Our all we are aware that this ABC efflux transporters is one of the culprit in bestowing the resistance to the cancer cells, because these transporters help in effluxing the drug molecules and not making available the drugs concentration inside the cells. And we have selected this MRP1, ABCG2 and PGP1 as these three transporters are more explicitly expressed in TNBC cancers. And here you find that these three molecules have differential, you just note that the differential regulatory effect of these flavonoids. It's the wrong notion rather that these molecules will have the same effect on regulation of the ABC transporters, but you find here that the MRP1 is downregulated by epigenin, while there is no effect of galangine on MRP1, and same is the case with all other three molecules. Here, interestingly, you will see that PGP1 is not at all hampered by this epigenin, galangine, and luteolin molecule. So this gives a cues that we need to have some pharmacophore addition to the scaffolds of the flavonoids for the effective inhibition of these transport molecules. We wanted to see as to, we have selected these two representative tumor suppressor miRNAs, MIR334 and MIR200B. The reason that we have selected these MIRs because they are involved and related with a lot of functions of the epigenetic modifications. And we have also selected this oncogene, which is also expressed in triple-negative breast cancer, MIR21. And here you will find that these molecules, which are taken further as promising flavonoids, they have very, very strong effect on inhibition of the tumor suppressors, and they have the upregulation of the tumor suppressor microRNAs, and they have significantly down-regulated the MIR21, which happens to be a oncogene. We have just initiated the efforts of cloning this MIR124, which is one of the important tumor suppressor MIR in triple-negative breast cancer. We have cloned it in R1 vector. Dr. Upasana has done this work, and we initially standardized this protocol. We just wanted to see that whether this MIR21 in cloning state, when we transfected this particular construct in the MDA23 cell ones, you will find that the MIR124 positive cells, as this particular MIR is cytotoxic, there is less fluorescence of this DS proteins, which is coupled with the CMV promoter. And here, in case of R1, where there is no presence of the MIR, which is only a vector, there is a better expression of this DS protein. And obviously, the MIR21 negative and R1 has no expression, as there is no presence of this. So we have also performed these experiments, and we just wanted to confirm that whether this same story happens at proteomic level or no. And here we find that the HDAC6, HDAC1 and HDAC3 molecules are being drastically, significantly down-regulated by this epigenin, galangine and luteolin molecules. We have also confirmed some of the effects of these particular flavonoids on the apoptosis, and you will find that the BCL, which happens to be a pro-apoptotic protein, has been drastically down-regulated in case of, as we all start the apoptosis regulation process with the cleavage of PARP, which happens to be an initial signature for the induction of apoptosis. And here we find that epigenin and luteolin have successfully cleaved these molecules. At the end, we just wanted to see what happens when we add these molecules, which have shown some impressive biological activities, when we take in combinations. We have used this particular SAHA as a standard epigenetic drug, and you will find that they have the states significantly, as it has been calculated by Chow-Kalli's equation, where it's a known index that if the combination index is equal to 1, it shows additive effect. If it is less than 1, it is synergism, and you will find that the ratio index is less than 1, and here is clear indication that the flavonoids do value addition in a synergistic mode with the SAHA. We have, in order to unravel the structure-active relationship and the molecular interactions with the several species of the HDAC, we have docked these complexes with these flavonoid molecules by using this gold score and skin score, and you will find that they have demonstrated significant scores, and they do have very perfect binding to these active sites of various species of HDAC molecules. We have measured all those HDAC bond angles, the distances, the molecules involved in that, and it's an entire scenario of that. There is a very concrete establishment of all those forces and bonds which per se are required for successful binding of the molecules to the active sites. We have performed the 100 nanosecond molecular dynamic simulation in order to ascertain that what happens actually to the thermodynamic at thermodynamic level, and you will find that the docked complexes, when they are subjected for molecular dynamic simulation, there is no perturbations, and the RMSD graphs and RMSF plots, which per se indicates the stability of these docked complexes, they are little bit. You will also find that here the hydrogen bonding remains consistent up to even the 100 nanosecond simulation. This entire scenario states that the binding of this epigenin, galangine, and this particular butylene molecules to HDAC 1 and 3 has sizable comfort zone in binding with the active sites of HDAC 1 and 3. And that has also been shown as what happens, the interactive modes of these molecules when they are under molecular dynamic simulations. So this is, again, we have measured the hydrogen bonding interactions with the simulated complexes with HDAC 1 and 3, as we have done this study with HDAC 1 and 3 because these two molecules are very instrumental in terms of regulating the progression of BSC cancer. So just a take-home message from our work that these molecules that we have selected for this apigenin, galangine, and deuterium are impressive in terms of showing the biological activities like they are demonstrated cytotoxicity, inhibited migrations, then they have also inhibited the cell cycle, they have induced apoptosis, induced reactive oxygen species, and more precisely they have inhibited that 1, 2, 3, 4, 5, 6, and 8, and when we have normalized these results with the gap pH. The protein expression levels of HDAC 1, 3, 4, and 6 were also reduced after treatment of these promising flavonoids. And this expression of DNMT1 and HMT downlimit after treatment of these molecules. Interestingly, this oncogenic MIR21 was downlimited after treatment of these two molecules, whereas MIR34 and 20B was significantly upregulated after treatment of these flavonoid molecules. The combinatorial effect is clearly demonstrated that these flavonoids do exert their effect by synergistic mode, and we wanted to see further, and we have analyzed this, confirmed this effect by this Western blot analysis, and we have also done some molecular docking and MD simulation in order to ascertain the thermodynamic stability of these docked complexes with that. So I would just like to concentrate your attention that the entire story of how dietary molecules exert their effect on in the cancer progression. I have recently wrote this book, which is published by the Springer Nature. This book has taken attention of the Daily Express UK London, and they have given one complete chapter of mine in the Daily Express. I invite you to please have a cursory look at the contents of the book, and some of the chapters may help you to make progress of your research. So these are my kits which we are working here. I had an opportunity to take funding from the SCRB, ICMR, and RUSA. Once again, I apologize myself to the organizers for making inconvenience in the last. I have experience of how difficult it is to have this organization. Thank you so much.
Thank you,
Professor.
Thank you, Professor
questions, I'll be happy.
Yeah, I think we are running short of time, so we would like to conclude the talk, and thank you so much for your